Thank you all for joining us. My name is Beth Chafin, and I'm the Science Communications and Development Manager at CCMI. And we're joined today by Dr. Gretchen Goodbody-Gringley, who is CCMI's Director of Research, and she's heading up our Vision 2025 Reef Resilience and Restoration team. Dr. Goodbody-Gringley previously was at BIOS, the Bermuda Institute of Ocean Sciences, where she led the Reef Ecology and Evolution Lab. Her research focuses on population structure, reproductive ecology, and genetic connectivity of a variety of organisms that inhabit coral reef ecosystems. And today, she's going to be sharing findings about the change over time for coral cover, composition, algae, and fish density and biomass from our 20-year data set of annual reef monitoring that took place on Little Cayman. She'll be discussing the results from those surveys and what they tell us about the stability of fish and coral populations on Little Cayman. And then she'll also, towards the end of the presentation, be briefly touching on Stony coral tissue loss disease, which has recently been confirmed to be on Grand Cayman. Just a little bit of admin before I hand everything over to Dr. Goodbody-Gringley. We're gonna keep everyone muted, and then if you have questions, we ask that you submit them in the chat box, and I'll ask them either throughout the presentation as they're relevant or at the end of Gretchen when we're ready to do a question and answer period. So we'll go ahead and get started. Gretchen, I've handed over hosting to you, so you should be able to now share your screen and share the presentation with everyone.
Great, thank you, Beth. And thank you everyone for joining us today to talk about 20 years of reef resilience on Little Cayman Island. So I just want to give a bit of background before I get into the data and what we have done thus far in Little Cayman. So we all are aware that coral reefs are intrinsically important to our island ecosystems. They provide support for the structural integrity of our islands, as well as economic benefits through fisheries support, tourism, etc. However, coral reefs are increasingly threatened by environmental impacts and anthropogenic impacts. These include things like physical disturbance from storms, disease outbreaks such as white band disease, overfishing, and of course, increasing temperature that has led to coral bleaching. And so if we look at some of the data that has been collected over the last several decades around the Caribbean, we've seen this trajectory of decline. So since about the mid-1970s, when coral reef ecologists really started monitoring the cover of coral on the reefs in the Caribbean, we have seen this steady decline, which has been linked to things like disturbance events. For example, the work by Peter Mumby here that I'm showing in this graph shows that one of the first major events to impact coral reefs occurred in the late 1970s with Hurricane Allen. And then subsequent to that, there were outbreaks of white band disease that specifically affected the acroporid corals. So those are our elkhorn and staghorn corals, which used to be the predominant species on the reefs throughout the Caribbean. And you can see that in this image here on the right, where you have this reef that was dominated by Acropora palmata, or the elkhorn coral, had overall percent coral cover of over 60%. And then post these impact events, you see that there's very little Acropora remaining. And in fact, today, the average coral cover across the Caribbean is somewhere hovering around 10%, which is a drastic decline from that initial 75% in the mid-1970s. So I mentioned these two acroporid corals as sort of these keystone species, and that's really those are the two species that we can really track this decline. And even a recent paper by Katie Kramer was looking at the prehistoric impacts or decline in these two coral species since the Pleistocene. And you can see that even prior to human impacts on coral reefs, there was some significant decline in these coral species. So it's possible that these environmental impacts were already affecting the corals in the Caribbean before we even noticed this outbreak of white band disease. But this white band disease outbreak that happened in the late 1970s, early 1980s, really was the main key factor that resulted in the loss of our structural integrity of the coral reefs throughout the Caribbean. And this was coupled with a disease that also impacted the Diadema antillarum, or the long-spined sea urchin, which you do see quite frequently around the Cayman Islands. And these two diseases acted in a synergistic way that resulted in the loss of coral cover and the overgrowth of macroalgae, and really changed the face of coral reefs. As we start to move forward in time, we begin to see the impacts of fossil fuel burning and our human impacts to the environment and really the atmosphere. As you can see, the increasing temperature in this graph on the bottom has started to now impact the prevalence of global bleaching events. So we did see the first mass bleaching event in the early 80s, but the first global event happened in 1998. The second was in 2010, the third in 2015, and we have seen subsequent major global bleaching events in 2016 and 2017. For example, in the Great Barrier Reef, if you're following this kind of research, you probably are aware that there has been extensive bleaching occurring over the last several years, recurrent bleaching events that have resulted in massive loss of corals throughout that system. So one of the ways that we can try to keep track of what's happening on our reef and make predictions about how our reef will respond to these types of impacts or future potential events is by simply tracking their health state over time. And so this is because the coral reef that we're looking at is not just a singular species. It's not only even composed of just corals. It's composed of a variety of different types of taxa, including corals, algae, a variety of invertebrates such as sponges, mobile invertebrates, and also the fish communities. And we know that when one of these things changes in the system, it can dramatically affect another component. And so we can see these sort of cascading effects. If we impact one component of our ecosystem, years down the line, we may be able to track that then resulted in a shift. And so by monitoring these various components of our ecosystem over time, we can start to see if small changes are occurring and what that might indicate in terms of our health of the reef that we have here, and also the resilience to future change. So CCMI has actually been monitoring this health state of the coral reefs at Little Cayman every year since 1999. And we're doing this using the AGRRA protocol. So AGRRA stands for the Atlantic and Gulf Rapid Reef Assessment. And this program actually began in, I believe, 1997. So CCMI was one of the early groups to be on board with doing AGRRA surveys on a consistent basis at our reefs in front of the lab at Little Cayman. And what we do is we assess the coral community, the benthic composition, and the fish communities at a variety of sites across Little Cayman using this standardized protocol. And that data can then be input into a larger data set that others are collecting across the Caribbean and used to make comparisons about how, for example, the reefs in Little Cayman are faring as opposed to the reefs in, say, the Florida Keys. And so this can become extremely useful in terms of management and conservation strategies as to, what are we doing differently than someone else, and how has that potentially impacted the state of our reef system. So in order to do these standardized protocols, we have a series of methods, and we didn't come up with these methods. This is actually what AGRRA has developed, and it does change slightly over time as we refine the protocols. And there's every year AGRRA sends out an updated version of the protocol that we will follow. And so to do these standardized protocols, we're looking for coral cover on the reef. And so to do this, we lay out a 10-meter transect line. and we count and identify every single coral that we encounter along that 10-meter transect, and we measure its length, width, and height. And then we assign that coral a health state. So is it perfectly healthy? Is it showing signs of a disease potentially? Is it showing indications of bleaching? Are there lesions from, say, parrotfish bites? So all of that is noted in our data sheet. The second thing we do is we assess what the composition is of the benthos. So what are the various components of that community? And so we assess, for example, what percentage of this small quadrat that we place down is macroalgae, what percentage is coral, what percentage are different kinds of invertebrates, for example. And then finally, we do fish transects. And so again, we use a 10-meter long transect line, and we swim along the transect and identify and count and measure all of the fish that we encounter along the way. And generally, we do between 6 and 12 transects for each coral and fish survey, and we do this at 6 to 12 sites around Little Cayman. The number of survey sites that we look at every year differs slightly, and this is primarily due to personnel or weather restrictions, for example, that may inhibit us doing as many sites as we would like to do. So if we just jump right into what these data are showing us, one of the key results is, of course, that percent coral cover. So this is just done by counting all the corals that we encounter, and then we standardize this for per meter squared, and then we can get the average percent contribution of corals across the various sites. And so you can see our data here. We have the year on the x-axis and the mean percent coral cover on the y-axis. And there has been quite a bit of fluctuation on an annual basis. So there does seem to be a bit of rise and fall in the percent cover of corals that we've seen over time. However, in general, there has not been a statistical decline in coral cover. So there is a bit of a trend towards reduced coral cover, in that we started out in 1999, 2000 at around 24% coral cover overall, and we are now hovering for the last 5 years or so around 20% coral cover. I do think it's really insightful, however, that we haven't seen any change at all since around 2013. So that does indicate that at least for the last 7 years or so, the coral cover is quite stable at Little Cayman. Do you have a question, Beth?
Hi, yes, thanks, Gretchen. So we do have a question from Jen, who wanted to know, when we're conducting these coral surveys, are we surveying the same sites? Are they fixed survey transects every year, or do we change and vary those sites as time goes on?
Thanks. That is an excellent question. So we do survey the same locations every year, but we are not using fixed transect lines. So the actual location of those 10-meter transects does vary slightly, but they are sort of at the same, we drop in at the same GPS coordinate site for each one of those locations every year. So even though we haven't seen a drastic decline in coral cover, which is great, we definitely don't want to see a decline in coral cover. Another indication that the reefs could be potentially impacted or are undergoing some sort of change is by looking at the composition of the different corals that are making up this community. And so we do this using our species composition, and we're looking at the percent contribution of different species over time. And what's quite interesting here is that even though we haven't found a change in overall coral cover, there is a significant shift in the dominant species that we're finding on the reef. So for example, in the early 2000s, the predominant coral species were the orbicelids. So these were Montastria cavernosa, Orbicella franksii, Orbicella annularis, Orbicella faviolata, or the star corals as they're generally known. These are large, massive corals. Some of these coral colonies can be, the size of a VW bug. They're huge coral bommies. And what's really interesting is that we have seen a major shift or decline in the predominance of those types of corals towards smaller, more plate-like and small boulder corals, generally in the genus Parrotfish or AGRRRA. And so this is quite a distinctive shift in species composition. Another thing that's interesting about this shift is that the orbicelids are broadcasting corals. So in terms of how they reproduce, they release sperm and eggs into the water column, and fertilization happens externally, and it can impact dispersal potential. So these larvae have 2 weeks in the water column and to repopulate other islands around the Caymans. As far as Parrotfish and AGRRRA, these are actually brooding species, and so that means that their larvae are fertilized internally, and generally they have a much more limited dispersal potential. So these tend to be more self-recruiting. And again, this is going to impact the overall resilience of our reef system if we are completely reliant now, or more so reliant on locally recruiting individuals. If something were to happen, for example, to a specific reef site, it would be less likely to recover than if we had some more orbicellas or broadcasting species present. So if we look at the cover of algae, we can actually see that there has been a significant decline in algal cover. So this is macroalgae. So all of the fluffy algae that we see on the benthos, the things like Dictyota or Lobophora, these have actually declined. And this is a great sign, and it does indicate that the reefs are quite healthy, that probably we're maintaining a healthy stock of herbivores, not just fish, but probably also urchins as well. And also indicates that the reefs in Little Cayman have not even begun to undergo any sort of phase shift towards predominance of macroalgae, which has been seen in other regions. So even as early as the 1980s, there was a lot of discussion about this transition from coral cover, predominantly coral cover, to macroalgal cover, and what we call a phase shift. And luckily, we don't seem to see that pattern happening on Little Cayman. So it does appear that the algal or the macroalgae is held under control, probably by a combination of herbivores and also low nutrient input, as there is very little human impact on Little Cayman. And so that brings us right into what we found with the fish. And so this is very good and exciting data. And what we're showing here is that there has been absolutely no change statistically in the amount or the abundance of fish that we are seeing in Little Cayman. And so I'm showing here in this graph fish density, which is the number of fish that we've counted, but we found the exact same result for biomass. So if we consider the size of every fish that we looked at as well, overall the size of the fish is about the same and the number of fish is the same. And so this is really important information, and it indicates that our management strategies, particularly with our MPA system in Little Cayman, is working, and we are in fact protecting our local fish populations. We can dive a little deeper into the types of fish as well, and look at how their abundances have changed over time. And sometimes this can get a little more detail, so we can tease out, for example, how are those, or how is the macroalgae in fact maintained at such a low level. So if we first look at the parrotfish density over time, we can see that in fact there is a statistical trend towards increasing numbers of parrotfish since 1999. So this is very positive finding, and it's probably one of the reasons why we are in fact seeing this steady decline in macroalgal abundance, because of course we have a healthy population of parrotfish, and parrotfish are this top herbivore in the system. So having this healthy population of parrotfish will likely lead to a stable ecosystem composition. Interestingly, when we looked at the biomass of the parrotfish, there was no change over time. And also, if we looked at the average size of parrotfish from 1999 to 2019, there was a slight decrease in the mean size of the fish. But because we're having this concurrent increase in the actual number of fishes that we're seeing, we think this might actually indicate that there is an influx of juvenile parrotfish into the system. And this could be very positive and indicate, again, that our management strategies on Little Cayman are really working well. If we start to try to delve into these correlations between parrotfish density, for example, and coral cover or algal cover, what we've done here is what we call a correlation matrix. So this is taking all of these various components in the data that we've collected and putting them into a model to see which ones correlate with which other things. And so what we found, for example, in this graph, anything that has an X over it is not statistically significant, and anything that does not have an X is statistically significant. So I just wanted to point out a couple things here. First is that we found a significant correlation between overall coral cover and parrotfish density. So this is indicating that sites with high densities of parrotfishes have a higher percent coral cover. So this, again, is probably one of those things that's really assisting our stability of the reef system, is this high density of parrotfish that we're finding at specific sites. And another correlation that pops out is the density of herbivores with the percent cover of macroalgae. And so you can see that overall fish density also correlates, as well as overall herbivore density. And so what this is indicating is that sites that have higher densities of herbivorous fishes are able to maintain a lower percent cover of macroalgae. And of course, this is intuitive and prevalent throughout the literature, but it's always nice to have it confirmed statistically that, in fact, our increased densities of herbivores do result in lower cover of macroalgae. Finally, we want to talk a bit about the Nassau Grouper, because there has been such a concerted effort in the Cayman Islands to protect the spawning aggregations through the Grouper Moon Project. And so when we looked at the overall biomass of grouper over time from 1999 to 2019, we in fact found a statistical increase in biomass over time of the Nassau grouper. Interestingly, the density did not change. So there was absolutely no change in the number of fish that we counted. However, the average size of the grouper that we were seeing was significantly larger. So this is basically indicating that the protection, again, is working, and we're seeing larger fish now on the reef. So they're making it to these later age classes, and probably also contributing more to the reproductive pool. So this is really great news, and it shows that the management strategy is in fact working for the Nassau grouper. So finally, if we take this all in together and look at the overall health state of the reefs in Little Cayman, here we've classified our reefs based on health state, and it's really just a measure of percent coral cover. So how many sites were, for example, 5 to 10%, how many were 10 to 15, 15 to 20, 20 to 25, or over 25% coral cover. And those were the boundaries for basically poor, fair, good, good plus, and very good. And what you can see is there has been, just like there's been a shift in the overall mean coral cover over time, there has been a shift in sort of the frequency of these health states over time as well. So in 1999, of course, we had the highest percentage of sites that were coming in as very good. And then we saw a decline in health state in the early 2000s, but it seems that the reefs are coming back slightly. So again, currently we're looking at reefs that are predominantly, the majority of reefs are either categorized as good or good plus. And so this speaks very highly for the health state of the reefs here in Little Cayman, and indicates that in fact the coral cover is relatively steady over time. So how does that compare with the rest of the Caribbean? And so if we look at the work published by Jeremy Jackson in 2014, when he was looking at coral cover across these various sites using a meta-analysis, you can see that the sites that he's noted here in the map that are highlighted in orange have undergone drastic declines. So by 1984, they were already at this 5 to 10% coral cover level. If we then look at the sites that are denoted with a blue square, you can see that they have also undergone a significant decline over time, but it was a bit of a slower decline, so it took longer to get down to this 10% coral cover. And then there are a few sites that are actually denoted with these green triangles, which have maintained relatively high coral cover since the mid-1970s. And those are, for example, Bermuda, Barbados, Bonaire, and the Flower Garden banks. And while we are not quite as high in terms of that overall percent cover, we are currently at 20% coral cover. And in comparison to the rest of the Caribbean, which is at roughly 10% coral cover, I would say that we are faring a bit better than the rest of the Caribbean. And especially the fact that our fish populations are stable and that our herbivorous fish populations have increased, as well as the biomass of our grouper species, our top predators. I think this all points towards the fact that the reefs in Little Cayman are in fact faring quite well and will likely be resilient over time. So as I just said, this stable coral cover over 20 years indicates that our reefs are quite resilient and have not undergone these drastic declines that we've seen in other parts of the Caribbean. And hopefully this puts us in a better position in terms of dealing with global climate change impacts, as far as, you know, looking into the future. If we have higher biodiversity, higher genetic diversity, because we have maintained this stable coral population, hopefully then the reef will be able to take on those global impacts as well. And likely as well, the stable fish populations further suggest a balanced ecosystem that will be resilient.
Gretchen, I'm going to jump in with a quick question here from Greg, going back to when you were talking more specifically about macroalgae. When we look at if particular sites have high macroalgal cover versus other sites, what are the potential causes of that, and does that decrease the health of the coral at that specific site?
Well, generally, there is a competition happening between macroalgae and coral. So if you do have a site in which the corals are impacted by some other stressor, then the macroalgae are probably going to take over that site. So it's sort of a war for space, for example, and oftentimes the macroalgae can grow much quicker than the coral, and so they have this capacity to overtake the reef. However, if that space is already taken up by a healthy coral population, it's unlikely that the macroalgae will overgrow live healthy coral, because corals do maintain this capacity to fight for that space. So they have stinging cells within their tentacles, and they will in fact sort of, what we say, wage war against another critter trying to infringe on its space. So, you know, as long as the overall system is balanced and healthy, then not the actual presence of macroalgae is not necessarily a detrimental thing to the coral. What becomes detrimental to the coral is if, for example, there's an impact and we have no herbivores there to maintain the macroalgae at a low level, and then the macroalgae are able to really overgrow that system. So again, it's all about this very delicate balance. And we do in fact find that some sites have much higher macroalgae than other sites, and there often is a difference. So usually sites that have a lot of macroalgae are not the sites that have very high coral cover. And so, you know, we do find this dichotomy between these two groups of organisms.
Okay, thanks. I think that's a great contextualization of what that is. Thank you.
Okay. All right. So, you know, overall, what we're taking away from these data are that we feel that the reefs on Little Cayman are quite healthy and hopefully resistant and resilient to future impacts and global change. However, having said that, it has been confirmed in the last few weeks that stony coral tissue loss is identified here in Grand Cayman. And so I wanted to touch on stony coral tissue loss disease just briefly so that you all are aware of what it is. And particularly if you see it, you can report it to the Department of Environment, as we are working towards tracking the outbreak and trying to find mitigation strategies. So stony coral tissue loss disease first popped up in Florida in 2014. And spread very rapidly down the Florida Reef track. It has decimated the Florida Reef tract, in fact, because it affects a variety of species. It presents very similar to another disease, which is called white plague. And so I think, initially it's often sort of pushed aside and said, oh, it's white plague. We're used to that. We see that every summer. White plague, however, is connected to increasing seawater temperature. So when it starts to get warm, usually we'll see an outbreak of white plague disease. White plague disease does not spread as rapidly. It generally doesn't affect as many species, and the mortality rate is not quite the same as with stony coral tissue loss disease. So stony coral tissue loss disease looks similar. It's basically a loss of the tissue. It almost looks as if the tissue is in fact sloughing off of the coral itself. So you can see from these images, in fact, all of these images were taken here on Grand this last week. So you can see that it affects a variety of different species and does have the potential for a whole colony mortality. So it's currently spread quite a lot around the Northern Caribbean. So for example, the US Virgin Islands has confirmed case, Jamaica, Mexico, and now here in Grand Cayman, in addition to where it was first noted in Florida. And the reason we want to just bring everyone's attention to this disease is because it spreads so quickly and it affects multiple species, and the mortality rate can be extremely high. So some of the first work coming out of Florida in 2016 by Bill Precht was denoting up to 100% mortality in specific coral species like the maize coral. So we want to bring it to everyone's attention so that if you do see signs of stony coral tissue loss disease when you're out snorkeling or scuba diving on the reefs in any of the 3 Cayman Islands, to please contact the Department of Environment and let them know where you have seen this disease outbreak. But we also want everyone to be reassured that we are working concurrently with the DOE. So CCMI and the DOE are working together to track the progression of the disease across the 3 islands. So the DOE really is spearheading this, and we are working together to try to look at rate of progression of the disease, and also collaborating with others in the United States at a few universities to try to identify potential pathogens. So we're taking samples of the corals and the disease sections to try to identify a potential virus or bacterial pathogen. And then we're also trying to develop some mitigation strategies to make sure that we are able to maintain these really vulnerable species, because there are 3 or 4 coral species that are more highly affected by the disease. And so we want to make sure that we are preserving that biodiversity and doing the best thing that we can to maintain the health of our reef. So with that, I think I'm going to leave it to questions.
Okay, great. Thank you, Gretchen. So just a question from Jen. She wanted us to post where they should report any potential examples of the Skittledy. So that should go directly to the Department of Environment, right?
That's correct.
There's also a Facebook page or a group that's called Cayman Islands Coral Watch. So I'll try to go on and get a link for that I can put into the chat box for everyone as well. There you go. I've got an email directly for Tammy at the DOE. Thank you, Katie, for passing that along. So I'm just going to post her email here in the group. If you've got photos and site information, please go ahead and send it to Tammy. Gretchen, Jamie asked a question about research that we actually just wrapped up for the last Darwin Initiative project on key herbivorous fish species. He said that at one of our recent lectures, one of the government officials mentioned an intention to protect parrotfish due to the correlation between herbivorous fish and reef health. And he wanted to know if there's been any further interest or effort to protect herbivorous fish. So I thought you could talk about the Biodiversity Action Plan and where that's gone.
Sure, yeah. So we did, in fact, again, we worked with the DOE and we wrote a herbivory biodiversity action plan that is now a chapter within the overall Coral Reef Biodiversity Action Plan. And we've made a few suggestions on how we should go about protecting key herbivores. Of course, parrotfish being the most important herbivorous fish species on the reef. But you might not really think about it, but also there are a couple other very important groups of herbivores, such as the chubs and the tangs, which often feed in large schools. And so if you're, for example, you may have seen these large schools of blue tangs inshore around Grand Cayman. They can really have a heavy impact. So it's not just the parrotfish that we're suggesting that we try to target protection, but also these other really important herbivorous species. But yes, that is still in the pipeline.
Great, thank you. Harrison did ask, is there going to be a publication produced from the data that you've presented today? I did share the technical report that we released and the summary reef report card in the chat box here. Is there any intention to do any further publication beyond that at this point?
Yes, we are currently writing the manuscript for publication, and we will have it available, or we'll certainly put it up on the CCMI social media broadcasts whenever that is available.
Okay, great. So Lola has asked how tourists can get involved when visiting our island and what they can do to support our efforts. So I will just start answering that by saying there are a ton of really easy things that you can do to reduce your impact on reefs, whether it's in your daily life, while you're diving, snorkeling, even, just when you're going to the store and at home. And we're actually sharing a lot of those resources through our social media. So I would recommend that people connect with us on social media, and I'm going to put the links to do so here in the chat box as well for our Facebook, Instagram, and Twitter accounts. And then Gretchen, if you want to come up with a couple of those specific suggestions that you could address right now.
In terms of what the tourists can do, obviously keeping an eye out for things like skittledy is going to be, the more ground we can cover with eyes in the water, the better. But also, of course, just like you said, reducing your impact. So really being great stewards of the environment, and when they're in the water, making sure they're not, touching things or leaving litter, those sorts of things. Ask for reusable straws. Little things do in fact make a difference. So as a collective whole, if we can all make these small changes, it does in fact have a large impact.
That is very true. I'm also
another thing also, if we can try to promote local fish versus imported fish, but even consuming sustainable fish populations by tourists like lionfish, for example, instead of grouper, is another great way that, even if they don't get in the water, it has a great impact on the reef community.
That is so true. That's a really good thing to point out. I'm also just going to drop a link in the chat box. We appreciate everyone participating today. We're really glad for your interest. Some of you have already made a donation when you registered to participate, but also if you would like to make a donation to CCMI to support our ongoing work, I'm going to drop a link in the chat box for where you can easily do that. We've got some amazing sponsors that have made this Reef Lecture series possible. So I do want to thank them. They're here on your slide. And also people can stay engaged. We've got another Reef Lecture coming up in September that I put a link for in the chat box as well. Gretchen, I'm going to ask you another question that I know you're going to be excited to address, which is from Jen about lionfish. She said, have you tracked the change in lionfish populations over the last 10 years since lionfish were first detected in the Cayman Islands?
We have worked a bit in the last few years, but doing surveys in Little Cayman. So for example, our former postdoc was working quite heavily on lionfish work and monitoring lionfish populations, but not over the last 10 years. And I would say that anecdotally, what we have seen is a general steady, if not a decline, in the lionfish population in the Cayman Islands.
And I do know that for people who participate in culling efforts throughout the islands here, that the DOE asks you to record and send in information about the size and gender of your catches. So there is some basic long-term data that is being recorded within the Cayman Islands. Okay, guys, I don't think that we have any other questions coming in. So with that, I'll just wrap it up and say thank you again all for participating. Thank you, Gretchen, for this presentation. Do you have any closing words to leave us with?
Just thanks everyone for tuning in, and stay tuned for more information from CCMI as we move forward, especially with regards to the stony coral tissue loss.
Great. Thank you all. Have a great day.